Okay.
We, starting with a keynote address, we are very excited to have Dr. Nicole Coufal here from University of California, San Diego. We heard a little bit about her work yesterday in the patient and family gathering meeting, which is very exciting. And as clinicians, we can all attest to that when we hear the word neurodegeneration, it gives us a little bit of anxiety, maybe a little more than a little bit of anxiety, because A, we do not understand this. B, of course, because of that, we don't know what to do with it. And it's not just a phenomena that is seen in LCH. Now we are seeing similar late onset neurological cognitive issues, even in adults with ECD later on, even if they are on targeted therapies and in remission, apparently. So Dr. Coufal's work has been focused on this rare phenomena, and she's going to share about her journey and about her work. And our goal is to see how we can apply that in our rare population, or also how we can collaborate with her. So please join me in welcoming her to the stage.
Thank you so much for the kind introduction, and I really look forward to hearing more from all of you as well. So I like to start by saying I'm not an oncologist. I'm actually a pediatric intensivist, so and my lab does very much neuroimmunology work, mostly in very basic mechanisms or in leukodystrophies and mitochondrial diseases. So this is a little bit outside of what my lab usually does, but we've been working on histiocyte disorders now for 5 or 6 years. Even closer, okay. For the last 5 or 6 years. So I'm really excited to tell you about our work, and I really look forward to your questions and collaboration, because we very much are a very collaborative group. So I don't have any major disclosures. I do some industry work in other situations or other settings. But I do like to start off by thanking the amazing people who do the work. The patients and the families. This was the patient with Langerhans Cell Histiocytosis. His mom gave me this picture after he got much better, who have donated to our work in terms of samples and blood samples, and the great team that I get to work with in the lab, because really the work is not only me at all, and so I like to put them up front and center. I don't think I need to talk about the histopathology. You probably all know it way better than I do, but I do like to point out that kind of black box that comes with neurodegeneration. And I think there's, more and more information about how different progenitors lead to different, at least in Langerhans and some of the other histiocyte disorders, different subtypes and different pathologies. But how neurodegeneration works, I think, is probably the most poorly understood of the kind of manifestations of histiocyte disorders. And so that's the thing that I got interested in and really wanted to try and understand better. And I think how those MAP kinase activated cells take on different phenotypes, interact with the other innate immune cells and the kind of brain in general, and how we can try to understand that better in order to have better treatments. I think the prevalence of neurodegeneration, every couple of years I look and it gets higher than it was the last time I looked. So the estimates vary really widely, but in ECD it's about 50%, it seems to, from the studies that I've read, and the presentations are really diverse. But there's a lot of ataxia, dysarthria, and it's an independent predictor of morbidity and mortality. And it's often treatment refractory or progressive. So I think while we're doing a really great job getting therapies into patients, only, from my reading, about half of them respond to some of the current treatments. So I think there's a lot of space here to understand this pathophysiology better and to treat it better. So these are some of the unanswered questions that Carl Allen posed to me years ago that we've been working on trying to provide some basic ways of looking at and thinking about to try and understand better. So what drives that histiocytic neurodegeneration? Why is it so variable? I think a lot of the studies that have come out have been based on lesional biopsies, and you have a lot of very many different cell types in them. Some of them carry BRAF V600E, some of them are infiltrating T cells, some of them are other cell types, some of them are perhaps other infiltrating monocytes. So how do you differentiate what comes from which cell type, and how do you decide what you need to treat? And why is there so much heterogeneity in the treatment response, and why is there recurrence after stopping treatment? So what are the treatment-resistant cells, the stem cell clones perhaps, that are leading to recurrence of disease, and how can we target those better? So how can we try to dissect this out? And I think there's a lot of limitations to the current models. So brain biopsies are pretty rare. Like I said, there's a lot of different cell types in there, making it hard to tell what's chicken or the egg. There are mouse models. There's actually several different mouse models. Most of them don't get neurodegeneration at all, so that really limits what you can study from the mouse model. The ones that do get neurodegeneration, the only one that does, it doesn't get the heterogeneity of the human neurodegenerative disease, so there's not these granulomas and other kind of heterogeneous lesions that humans get. And then innate immunity in mice varies hugely, so those in the oncology sphere know that we very much in the scientific community use humanized mice to try and understand leukemias and lymphomas. You don't use the mouse immune system because it's so different, so trying to use the mouse immune system to understand histiocytic disorders, I think, has limitations. So I took this as we need a human model. And so that's a big part of what my lab has worked on the last 5 or 6 years. And so I'm going to tell you this on a kind of overview, how we tried to make a humanized model using induced pluripotent stem cells, using patients, what sorts of phenotyping we've been able to do in vitro to try to understand what the pathophysiology is of histiocytic disorders, and then how we tried to understand neurodegeneration. So for those of you who aren't familiar with induced pluripotent stem cells, this is taking samples from patients. In the old days, it was skin biopsies. Now it's much more commonly a blood sample, using a variety of things in the dish to try and turn those blood cells into primitive cells or induced pluripotent stem cells that can make any cell type of the body, so they can make all of the different germ layers, but from a dish, and try to use those to make the cell types of the brain that you can't easily get from patients, and be able to make neurons or microglia or other cell types and be able to look at how they respond to therapies or their phenotypes, and hopefully preclinically test therapies. And so we did this from a pretty large cohort of histiocyte patients. We did this from 10 patients in total. The first one was one of my patients, and then Carl Allen helped me find other patients, either from their leftover bone marrow samples or from peripheral blood. And it was all patients, let's see if I have a pointer. So it was all patients who carried the BRAF V600E variant somatically, where they had some in their peripheral blood that we could try to then make induced pluripotent stem cells in a dish, get a mosaic, and try to get from the same patient both stem cells that carried the BRAF V600E mutation and those that didn't. And so in that way, to be able to have cell lines that have the same genetic background that put them at risk for histiocyte disorders, and have both the ones with the pathogenic variant and those without. And because this is a little bit shorter talk, I won't tell you the trials and tribulations of this, but this only worked for one of the 10 patients, and so we actually ended up using CRISPR-Cas9 gene editing to induce the BRAF mutation in the patient-derived cell lines from some of the other patients in order to have a pretty good cohort. And so what we wanted to do, and I think part of that is just the heterogeneity that you have sometimes, like what exact progenitor in the blood is able to become a stem cell is not clear, but because you can have BRAF in so many different cell types within the blood, that by chance you didn't get the right thing. And so what we wanted to do was to make the cells of the brain, the innate immune cells of the brain, microglia, from these induced pluripotent stem cells, and ask, what happens and how does that contribute to neurodegeneration? And the differentiation, it takes about 6 weeks, so everything with stem cells is slow and takes a really long time, and a lot of different cytokines to try to push them to the correct cell fate. And so what we did, what we started with was doing RNA-seq, so doing transcriptional analysis and asking, using a cohort of those that were generated from the patient where we got both BRAF V600E and those where we induced it through CRISPR-Cas9 gene editing, what happens when you have this V600E variant in brain myeloid cells. And what you can see here on this PCA is that when you make these, we call them IMGs, or induced microglia from induced pluripotent stem cells, they look more like primary human microglia, either from neonates or from adults, than they look like other kinds of monocytes. So I think that what you're making in a dish is reasonably close to a brain myeloid cell. This is the hematopoietic progenitor, and what you can see here is that those from patients that carry the BRAF V600E variant are distinctly different than the other ones. It's not that you're making a completely different cell type. You're not making a CD14 monocyte or a CD16 monocyte. And then the ones in gray here are the ones from the isogenic controls, mixed in blue with ones from other studies that we've done. So they're quite distinct. And what we see on the differential gene expression is a lot of inflammatory pathways, especially, are elevated in the BRAF V600E. And so we wanted to ask, what are these genes that are higher in the V600E brain myeloid cells, and how does is that relevant to histiocytic disorders? I think that's really important anytime you're doing something in a dish, is to try to then correlate that to something from a patient. Does it matter? So these are the gene pathways that are elevated in the V600E brain myeloid cells. You can see MAP kinase is up here. There's a lot of inflammatory terms. So these are very inflamed macrophages. And we wanted to compare this data set to one from primary patients. And so the data set we compared to, this is whole brain RNA-seq from several patients who had 3 different histiocyte disorders from this publication. So this is not from their isolated brain myeloid cells. This is whole brain RNA-seq. And you can see that the overlap is tremendous on the heat map. So the kinds of things that are dysregulated in this model in a dish very closely replicate what happened in the patient. And so I think this is actually one of the highest correlations I've seen, having done this for a long time. So I think this is a pretty good model. And so we wanted to ask, if we look at these in a dish, what can we say about the progenitors? Because a lot of these histiocyte disorder mutations are occurring in some blood progenitor, and then the more differentiated cell types that carry the V600E variant. And so we started with the hematopoietic progenitors. I think 2 things kind of came out of this. One was that they are more proliferative than the isogenics, the controls from the same patient. So you do have this smoldering where they're growing a little bit faster and are more proliferative than they would be otherwise. And when we fully differentiated these into brain myeloid cells, what we saw is that they're also apoptosis resistant. So there's a very kind of angry inflammatory cell type that is resistant to apoptosis. And so we looked at both cytochrome C release, which was higher, but then we did a growth factor withdrawal assay, and so we removed the growth factors that usually microglia are dependent on, and what you can see is that with growth factor deprivation, you get this huge death of normal microglia. If you give a CSF1 receptor inhibitor, microglia die. And what happened with the BRAF V600E myeloid cells is that they didn't care. They actually made their own CSF1, so they're quite apoptosis resistant. And so we think this gives us some good kind of cues moving forward about how to think about drug sensitivity. The other thing we wanted to think about is what is released by these in terms of cytokines, and how does that relate to biomarkers, and how does that relate to kind of some of the patient studies. And so this is cytokine data looking kind of high multiplexed cytokine release, and what we see across cell lines from multiple individuals. And what we see here is that some of the biomarkers that have been identified from histiocyte patients are also increased. This is just in a dish released into the media. So this is osteopontin. This is Dr. McLean's work. You can see osteopontin was specifically high in histiocyte patients in terms of their CSF, as well as some of the TNF-related pathways. And then metalloproteases have also been identified in a number of the mouse models. So we, again, think this correlates well with what comes from patients. And so what we wanted to do next was to ask, okay, we have all these cell lines. How can we do a humanized kind of brain model? This was our first attempt at organoid co-cultures. And so what we did here is make what you would call a mini brain in a dish. It's neurons, astrocytes, maybe oligodendrocyte precursors. There's no myeloid cells in here because they come from a different lineage. So we introduced from the same patient either microglia that are from the healthy genotype or the BRAF V600E, kind of co-cultured them and asked, okay, what happens? Do they cause neurodegeneration, and can we understand something about how that happens? And so what we saw in doing this broadly is that they really do cause changes in terms of what types of cells are within the organoid in the single cell. And so we see that proliferation here, and that the BRAF V600E have more microglia or brain myeloid cells because of that kind of proliferation. We see increase in the number of astrocytes, which are also supportive cells that are downstream, and a loss of some of the progenitor cell types that you would normally see. And so what does that kind of mean on a larger scale? Is we put together a reactive astrogliosis signature, so that is how the healthy normal astrocytes are reacting. Are they having an inflammatory reaction to the microglia? You can see the V600E do have this reactive astrogliosis signature that's increased, including a number of markers within the astrocytes indicative of increased inflammation. And then we also see neuronal death. So there's an increase in caspase-3 within the neurons. So I think the limitation of doing these organoids is that there's no vasculature, there's no myelination. They're really very primitive in a lot of ways. And so one of the things my lab's worked on for the last almost decade now is trying to come up with humanized mouse models to be able to ask questions about how human innate immunity causes neurological disease broadly, and then apply it to this specific question. And so this is a fiendishly difficult mouse model, but basically it's B and T cell depleted to allow for xenotransplantation to put human cells into a mouse. They also are humanized for CSF1, which is essential for microglial survival, and the mouse doesn't hit the human receptor. And then they have this CSF1 fire deletion, which is an enhancer within the CSF1 receptor that depletes all of the murine microglia. So basically, this is a healthy brain, it's a healthy mouse, and they have no microglia whatsoever, so the niche is completely open to put in human myeloid cells of whatever varietal and ask what they do. And so we've used this kind of extensively in a number of mitochondrial diseases to ask how mitochondrial diseases cause ataxia, but in this setting, what we can do is say, like, okay, when you put in the BRAF V600E brain myeloid cells, what does that do to the brain, and how does that work, basically? How much of the phenotype in histiocyte patients is specifically due to the myeloid cells themselves, without T cell infiltration, without interactions with the endogenous microglia, and then how can we try to kind of bring this closer and closer to what patients look like? And so we did these xenotransplantations, and what we found was that the BRAF V600E myeloid cells they're highly dysplastic. They're very angry. You can see this is what a healthy microglia tile across the brain. This is the same patient's healthy ones versus the V600E. They tile across the brain, they touch each other, they modulate synapses, and they don't normally kind of grow unfettered, as such things go. So you can see there's a huge number of increased cells, and they're these very small, angry, ramified cells. And so we wanted to look and see if some of the markers that are seen in patients with histiocyte disorders and neurodegeneration specifically are also seen here, and how much of, given that we've put in, this is a clonal cell line, it came from one cell originally, how much of this heterogeneity comes from just that one clone? And so what we saw was that some of these markers that are seen in patients, and I've put one of the patient pictures here on the side, are also seen, but not in every cell, so that this heterogeneity persists even from this one clonal kind of myeloid cell type. What we're grafting is basically a progenitor, a myeloid progenitor. And so we see that increase in CD68. We also see increases in antigen presentation in HLAs and in CD163, which is one of the hallmarks of a lot of the histiocyte disorders. And that was kind of massively increased throughout the brain. But we also saw this heterogeneity. So some things were homogeneous and some were very heterogeneous. And we found this really kind of interesting, because this is not something you see in any of the mouse models whatsoever, but is very characteristic of patients. And so versus all of the myeloid cells carry the V600E, you only see some cells that have phosphorylated ERK. We don't see this high expression, which is also true in many of the patients. We see increases in senescence markers. So this is beta-gal and P21, which is also kind of what Carl Allen has been reporting in the one mouse model that does get some neurodegeneration. And even though there's increased proliferation, it's small. So many of the cells, they kind of populate the brain, become this very angry inflammatory cell type with only small pockets of progenitors. And then we saw these small granulomas, which are also very characteristic of what patients have, which is that there's these, in here, put one of the patient ones in here, which is that there's these almost nodules or granulomas of antigen-presenting, EBA is a myeloid marker, This is a human nuclei marker. And then there's occasional, very, very rare patches of CD1A or CD207, but most of them are negative for those markers. So all of this kind of suggesting there's some genes by environment interaction that's leading to this heterogeneity and this pathology. And then we looked for damage to the brain, so we wanted to know how much damage are these cells causing, and does that also kind of replicate the phenotype of what we see in patients. And so there are increases in astrogliosis marked here. So this is an astrocyte marker, and GFAP is a reactive astrocyte marker. These large increases within the astrocytes for GFAP suggestive of astrogliosis. And we also looked for myelin loss and saw kind of global areas of demyelination and response. So I think a lot of this replicates the pathophysiology. And then we looked for neuronal death, and we wanted to see this kind of in two different settings. One of them was in, here, this is within the cortex. So we see, especially in layer 5, which is the large projecting neurons, so the most metabolically active and the most kind of sensitive to stress, decreases in the V600E, so loss within those. And then we also saw within the Purkinje cell layer and the cerebellum, neuronal death. So considered like what you would see with ataxia or some of the really prime features of histiocyte disorders. So this is the picture of the cerebellum, and you can see these are the Purkinje cells in a control engrafted animal, and this is a V600E, and you see kind of the loss of myelin, the loss of healthy Purkinje cells. There's only a couple left here, and this is also consistent with what's been seen in histiocyte patient brains. Lastly, what does this actually do to the mouse? Do they actually get symptoms of from all of this kind of inflammatory phenotyping? And what we saw was that they really do. And so we tested this a couple of different ways. The first one is the rotorod. They have to try and stay on the rod and not fall off. So it's a measure of motor coordination. And so they fell off much faster. They couldn't stay on the rod at all. And the second one was one of limb clasping. So a normal mouse will kind of put its legs out so that if it drops, it will catch itself on the ground. And so they have this clasping phenotype where they've lost their normal motor coordination. We did a gait analysis, so you can see this is what the normal paw prints would look like of an animal. And so they have a wider gait and are less coordinated than they would be otherwise. And then we looked at survival. So we're introducing these pretty early postnatally. And you can see that they only survive out to about 2 months. So again, this is with 100% of their myeloid cells carrying BRAF V600E. I think the next steps for us have been now doing a mixture. So all patients are a mixture of healthy myeloid cells plus some percentage of V600E. This is a model that takes a long time. So we did 50-50, 90-10, and 99% healthy and 1% V600E. And all of those survived much, much longer. So healthy myeloid cells were protective. And even in the 99-1, we were surprised when we looked at the brains when they looked healthy. They had really large patches of V600E cells all through the brain, but having healthy myeloid cells was quite protective phenotypically. And so I think what that interaction is between your normal healthy myeloid cells and how those manage to limit disease is one of the things we want to follow up on next. All right, so what I've told you about today is that we've taken these histiocyte patients who have BRAF V600E mutants and healthy cells from their peripheral blood, generated a model where we can try to study this in a dish and ask some questions about what are the phenotypes of the progenitors, so how those progenitors are sensitive or not sensitive to drugs, how they proliferate, what happens when they're fully differentiated within the tissue environment, and how that can kind of relate to the symptoms that are seen by patients, how this replicates some of the biomarkers and patient-specific phenotypes, and how we can try to look at how they interact with the brain environment. And what we see is in the future is really now trying to ask more clinically relevant questions. I feel like we've really done a lot of diligence on trying to correlate this with primary human data, correlate this with pathology and with biomarkers. And so the ways we're moving this forward is really trying to ask questions about treatment sensitivity. We've started by using the hematopoietic progenitors and asking if we use BRAF inhibitors, MEK inhibitors, CSF1 inhibitors, how sensitive are they to those compared to the same patient's healthy myeloid progenitors, and then how does that work in terms of the more differentiated cell types? Are there certain stages that are more or less sensitive to drug treatment? How do we think that that works in terms of what are the treatment-resistant cell types that are leading to recurrence? And how can we try to kind of leverage this system to ask questions about new therapies? We started working with Dana-Farber Center on trying to do this with some of the senolytics and ask about BH3 profiling. And so how do we first start with the dish model and ask how can we ask questions about either single or combination therapies and what we think might work the best? And then how do we now translate that into the preclinical model, especially in our mouse model? And first starting with making this a more patient-realistic model by having mostly healthy myeloid cells and then having a small percentage of these BRAF V600Es. We started with mixed engraftments, and now we're moving towards adding in the V600E a little bit later in life, since there's a lot of debate about how that works ontologically, what is kind of the best model for how this works in patients, and then trying to figure out, because you can't test 40 things in a mouse population, what are the most relevant questions for patients in terms of preclinical modeling? Is that to do that with a BRAF V600E inhibitor, take it off and see what the cell types are that are leading to recurrence? Is that doing combined therapy? Like, how do we kind of best answer important preclinical questions? I think we've been really interested because it does seem like there are some CSF1 receptor trials out there that it seems like having healthy myeloid cells is protective, and they're much more dependent on CSF1 for their survival, that those may actually be harmful. So we've asked about whether we should be asking that question. And so those are some of the ways we're thinking about doing this as we kind of move forward and try and ask like really clinically relevant questions. I have a ton of people to thank. I did so the paper actually just came out 2 weeks ago. So in terms of setting up this model, like I said, that was really kindly supported by ECDGA. And we were so grateful. And this is my wonderful team that I work with. The paper took a long time, so we're halfway through with the next one. So I look forward to being able to tell you about those. And please, we're super collaborative, and we really want to do something that matters for clinicians and for patients. And really make it meaningful and impactful. So thank you so much. I'm so happy to take any questions.